I'm so excited. This is an event we've been trying to organize for a while now and finally decided to just give up and do it virtually. To begin, customarily at UBC, we acknowledge that the UBC Point Break campus is located on the traditional, ancestral, and unceded territory of the Musqueam people. My home, where I am currently working, is also on this unceded land, and I feel privileged and grateful to be here. Because we're scattered at the moment, you might be calling from the traditional territories of other peoples. So we invite you to learn about the traditional territories you are on by visiting native-land.ca. For accessibility reasons, I'm going to describe my physical appearance. I'm wearing a navy blouse. I have graying, short black hair, and I wear glasses. And behind me is the roof peak of my attic study that I love. We've sent around the respectful space statement and posted it in the chat. Thank you for your help in making this a safe learning space. Yeah, you already know we're being recorded. Okay, and you can turn off your video and microphone if you don't want to be recorded. For organizational purposes, please type your questions in the chat as we're going along, and you'll also have an opportunity to raise questions at the end. So today's event features two incredible scientists. First, our own President Santa Ono, and then Oxford University's Dr. Dianne Newbury. And this event is jointly sponsored by UBC Language Sciences and the Social Exposome Research Cluster. The Social Exposome Cluster, led by Dr. Michael Kobor, aims to understand the biological mechanisms by which social factors get under the skin to influence child health and development. UBC Language Sciences, led by Dr. Brian Jick and me, Janet Worker, facilitates collaborations that enable groundbreaking advances on how language shapes our individual and collective human experience. Okay, President Santa Ono, Dr. Santa Ono, is the 15th President and Vice Chancellor of the University of British Columbia. A superb scientist, a great president for all of us, a cellist, and just a wonderful human being. Installed as President and Vice Chancellor in 2016, he also serves as chair of the U15 group of universities. He's on the board of directors of Universities Canada, past chair of Research Universities of British Columbia. He's also a member of many boards, including Fulbright and MITAX. He's served in many other leadership positions, including the boards of the American Council on Education, the Council on Competitiveness. He's Chief Innovation Officer to the province of British Columbia. And prior to his appointment at UBC, Dr. Ono was president at the University of Cincinnati. He's a molecular immunologist educated at the University of Chicago and McGill. Dr. Ono has taught at Johns Hopkins University, Harvard University, and University College London. He's advised national and regional governments on education and mental health, another one of his priorities. And he's also advised companies such as GSK, Johnson & Johnson, and several others. He's on a number of, or has served on a number of editorial boards, including Immunology, the Journal of Biological Chemistry, and several others. He's a fellow of the American Association for the Advancement of Science, the Canadian Academy of Health Sciences, the Royal Society of Canada, the National Academy of Inventors USA, and the Johns Hopkins Society of Scholars. He holds honorary doctorates from Chiba University and the Vancouver School of Theology, and is a recipient of the Reginald Wilson Diversity Leadership Award, of which I think he's particularly proud, among several other awards. Dr. Ono did some of the groundbreaking work characterizing the gene underlying some of the neurodevelopmental disorders Dr. Newbury's research focuses on. It's thus with great pleasure that I present first Dr. Ono, who will speak about his work on this gene, and then he will introduce Dr. Newbury, who will then share her work with us. We're delighted to have both of them here today.
Oh, Janet, I didn't know I was going to speak about my research. If so, I would have brought up some slides, but I can talk just generally. about it. But thank you, Janet, for that introduction. And Michael, it's wonderful to see everyone, even though it's virtual. And I'm really looking forward to Dianne Newbury's talk. And I've really enjoyed following her progress over the past couple of years, and was really excited to hear of her discovery. I'll just give you sort of some background, and obviously Dianne knows about it because she works on the gene product encoded by this gene. But it was all about 30 years ago that I came across this gene that encodes a protein, which I called Nfx1. And it was called that because I was studying at the time a set of genes that are responsible for regulating the immune response. Those are called class 2 major histocompatibility complex molecules. And there are 3 that are encoded in the human genome on chromosome 6, DR, HLA-DR, HLA-DQ, and HLA-DP. And we had discovered that the 3 isotypes of these molecules in man were usually coordinately regulated, but occasionally the regulation of the DR and DP isotypes was regulated independently of DQ. And we wanted to try to understand the molecular basis of this differential expression, and to perhaps understand what the different roles of these 3 class 2 major histocompatibility complex molecules would be in the immune response in health and disease. In the process of trying to understand this differential regulation, we mapped the proximal promoters and also the enhanceosome of the genes that encoded these heterodimers, each isotype being encoded by an alpha and a beta chain that would fold together into a heterodimer that would present antigen to the T cell receptor on lymphocytes. And in mapping the enhanceosome and the proximal promoter of these genes, we found 2 critical elements: an inverted cat box, which was close to transcription start site. Then further upstream, there was something which we call the X box. The X box would then be further subdivided into 2 cis elements. One we called the X1 box and one we called the X2 box. The X1 box was a novel cis element. The X2 box looked something like an ATF or Krebs binding site, 2 transcription factors that would become defined as leucine zipper proteins, as you know, are one mechanism that heterodimers occur to create different sets of transcriptional activators on many eukaryotic promoters. So the X1 box is something that we really focused on. And at the time, we used expression screening of libraries to try to identify proteins that would bind sequence specifically to this new cis element called the X1 box. And it was through that we isolated this gene encoding a novel protein I called Nfx1, thinking that there might be additional X-box binding proteins identified as we carried on our research. And when we sequenced the gene, it ended up being quite an interesting DNA binding protein. At the time, it was one of the relatively new metal binding proteins that through iron in metals like zinc could fold in the work of other groups form zinc finger proteins that would bind sequence specifically through those elements. And NFX1 had a reiterated motif with multiple what we would call NFX fingers that were in the central part of the polypeptide. So it turns out that its expression is relatively ubiquitous in many cells, but what we found was it was inducible by a cytokine called interferon gamma, and that it seemed to repress both the human and murine forms the expression of class 2 MHC genes. The reason why that we hypothesized might be important is that it might regulate the duration of the immune response and might be important in controlling immunity against self, because these class 2 major histocompatibility complex genes were some of the most strongest determinants for susceptibility to autoimmunity. And so that's how NFX1 was isolated. And we studied both the human and murine genes encoding this interesting transcription factor. And I think, as all of you would recognize, what happens in 30 years is truly remarkable, and something that can't be predicted when you first look into a black box and look at a new kind of a protein. What's happened since is that it appears to be quite multifunctional, and there is not just one, but there seems to be paralogues of the gene. And it's been subsequently implicated in a ubiquitination pathway, and also in two diseases, Huntington's and Parkinson's disease. And I can tell you that back then, 30 years ago, we had no clue that the Nfx1 gene or its paralogue would have any role in something as exciting and fundamental as language acquisition. And that's what appears to be the case. And according to Dr. Newbury's breakthrough research, I find her findings exciting because I think language acquisition is something that's terribly exciting and certainly a strength of the research here at UBC. And so I think it's wonderful to have her here to talk about her research on the paralogue of Nfx1, something called Nfxl1 that looks very similar to Nfx1. So let me just stop there. That's a little bit about the gene, how it was discovered by screening an expression library, how it was characterized biochemically, how it was implicated first in limiting the immune response and functioning as a transcriptional repressor. But after 3 decades, you can see that this very interesting protein appears to have other roles in health and disease as well. So let me tell you about Dr. Newbury. And she's a molecular geneticist who studies genetic contributions to childhood neurodevelopmental disorders. Her investigations specifically focus around speech and language impairment and its relationship to disorders such as dyslexia. As she explains on in her publications, this research is important because it will help to understand why some children have language difficulties and what brain processes are important in language learning. It may also help to identify new kinds of language disorders and will clarify the relationships between language impairment and other developmental disorders. She's a senior lecturer in the Department of Biological and Medical Sciences in the Faculty of Health and Life Sciences at Oxford Brookes University. If I recall, that's relatively close to Oxford University, where her lab is based. And her work is funded by the Economic and Social Research Council and the British Academy. So I'm delighted and honored to introduce Dr. Dianne Newbury, somebody who is fascinated with this same molecule that I was lucky enough to come across 30 years ago. Welcome, Dianne.
Thank you. Thank you. Let's see if I can share my screen with you. Okay. Yes, and I can still see some of you. Okay. Can you all hear me okay? Perfect. Okay. So yes, I'm Dianne Newbury. Good evening from the UK, from a sunny day in Oxford. Good morning to those of you in Canada, and good afternoon for anyone in between. This is Oxford Brookes University here, and this is where I work. It's just outside of Oxford itself. Okay. Oh, let's see. Okay. Can you see my pointer on here? Or, yeah. Okay. So as President Ono said, in my research lab, we use genomics to look at the genetic contributions to speech and language. The kind of broader questions that we're interested in are things like, is language just a form of higher cognition, or is it something separate at a biological level? But mostly, do genetic mechanisms overlap between spoken and written language? And interestingly, what are the links between spoken and written language in terms of molecular bases? And what factors, this is the million-dollar question, what factors mediate positive outcomes in children who have language difficulties? Okay, so I just want to start. This is where the research that I'm going to be talking about today starts. It's this small island called the Robinson Crusoe Island, which, if you look on Google Maps, is positioned here. That's not very informative. So here's a zoomed in version. This is the shape of the island. It's a very small island, about 700 kilometers to the west of Chile, actually to the west of Santiago here, in the South Pacific Ocean. It has very limited access even today. You can see it's a very small island. There are aeroplanes, light aircraft that you can take in the summer months, but in the winter months, the only way to access it is across the ocean on a ship that goes to the island about once a month to deliver goods for the islanders. So it's a very, very interesting place. And here's a brief history of some of the things that have gone on in the island. So it was discovered in 1574 by somebody called Juan Fernandez, and it's part of the Juan Fernandez archipelago of islands off the coast of Chile. It's called the Robinson Crusoe Island because allegedly in 1704, Alexander Selkirk was sailing through the Pacific Ocean on a boat called the Cinque-Port, and he was not convinced that this ship was seaworthy. And so he said to his captain, I'd like you to drop me off. The next time we see any land, I would like to get off this boat because I don't feel safe at all, which his captain duly did. And this is Alexander Selkirk here. He stayed on the Robinson Crusoe Island for 4.5 years in complete solitude and survived on the island. And this is allegedly who the story of Robinson Crusoe, Daniel Defoe's famous story, was based upon. So if you go to the island today, you can visit Robinson Crusoe's cave. This is a cave in which he was reported to have lived for those 4.5 years on the island. And this here is a treasure hunter called Bernardo. And he is looking for Armada treasure on the island around Robinson Crusoe's cave, which he says is buried on the island. He started looking for his treasure about the same time that we started looking for genes. So we like to think it's kind of a parallel research strand to ours. So the treasure that he's looking for is the Treasure of Veracruz, which was left on the island by buccaneers shortly after Alexander Selkirk had been rescued from the island. Because of its position in relation to South America, it's been colonized various times over history because it's quite an important naval position because of where it is. But throughout history, it's been colonized, and then there have been natural disasters which have stopped the colonization of the island. So in the 19th century, it was used as a prison island. They would drop the prisoners off on the island and essentially leave them there to get on with their lives. But there was an earthquake and that was abandoned. And there have been earthquakes throughout history. So there have been various kind of colonizations throughout history. But then these events which have prevented people from living on there permanently. That is until 1877, when the island was bought by a Swiss baron called Alfred van Rott. Here he is. And he bought the island from the Chilean government to set up a lobster catching colony. So these are the lobsters that are still caught off the Robinson Crusoe Island today, and they're shipped back to Chile. These are some of the best lobsters in the world. And Alfred van Rott said that he bought the island particularly to fish for these lobsters and to ship them back to the mainland. That was in 1877, and the people who colonized the island then are the same group of people who live there today. So this is the longest colonization in the history of the island. As I said, it's still a very hard place to get to. So this is the airstrip here on the island, and this is the aircraft that will travel to the island in the summer months. But in the winter months, there's just a monthly ship that drops off provisions to the islanders because crossing by airplane is just too dangerous. I think there are very few places in the world that remain this isolated today. They do have television, they do have satellite communication, but that's patchy at best. It's a very hard place to get to. But nonetheless, this group of people still live on the island today. So the current population in the last official census, there were 633 residents who lived on the island. They all live in a single town. You can't actually see it here, but it's just up the hill on this picture here, which is around the bay, and primarily they earn their money from catching lobsters, which they fly back to the mainland. So there's one single town, there's one single school, but the children and the residents must attend the mainland if they want to go to the doctors, the dentist. When the children go to the high school, they tend to go to the mainland to live with relatives in Chile. In 2010, so it's still a very dangerous place to live. In 2010, there was a tsunami. This is what the bay looked like before the tsunami, and this is the bay after the tsunami. And you can see that all the buildings were just completely washed away by the tsunami. These were the municipal buildings down in the bay here, and luckily the tsunami hit in the evening, because if not, the school and all the hospital, all the municipal buildings were down here and were completely washed away. And these are currently being rebuilt further up the hill. This is the brave girl who sounded the tsunami alarm when the tsunami hit in 2010 and saved the lives of a lot of islanders. And these are some of the new buildings coming. So lots of the new buildings are in these containers, which are delivered on the island, and then they move them up the hill and use these to make new schools. This is a new school here, which is in a container. Okay. And there's a lot more information about the island, the way of life today, on this website here. Okay, so we're interested in these islanders because they are what we call a founder population. So a founder population is a population that's derived from a relatively small number of individuals who are closely related to each other. And as geneticists, these are really interesting to us because they have a reduced genetic variation, which means that they can offer increased power to detect contributory genetic variations because they have a much reduced genetic variation. So the genetic variation, as I said, this is just a recap. As I said, the island was bought in 1877 by Alfred von Rott, and it's said that he took 64 settlers to the island with him. And 30 years later, there were 122 people living on the island, derived from these 64 settlers. And as I said, most of the 600 people who live on the island today are still related to these 64 settlers. So the official historical documents say they were primarily Chilean families who settled on the island. But as I said, Alfred von Rott himself was Swiss, and he brought along some of his friends and family. So there was a Frenchman, an Englishman, a Portuguese, 2 German, 2 Italian families, and 13 Chilean families. But there are alternative accounts of how the most recent colonization of the island occurred. So Severin said, actually, the current settlers date back to slightly after this most recent settlement, whereas Woodward has an alternative account which says the settlers came about 10 years earlier, and Alfred von Rott then just arrived after these settlers. More recent publications from Pia Villanueva, who is our collaborator in Chile, said that the current residents on the island are descended from 8 founder families, and about 77% of the current population have surnames who are shared with the founders. They looked at the shape of the incisors and teeth and used these to try and map back the population. And that research said that about 5% of the islanders had indigenous ancestry, so Chilean indigenous ancestry. So there are various accounts about the current population. They are no doubt a founder population, but the actual history of the population, this is the best guesses that we have. So we've been able to use some of the genetic information from these islanders to map out some of that historical information. So we can use genetic markers, which are passed down from mothers to their children, and these are known as mitochondrial markers. And these are used throughout all kinds of genetic studies to map population movements around the world. So we can look at similarities and differences across these genetic markers to look at different populations. And similarly, we can use markers on the Y chromosome, which is passed from males down to their sons. So we looked at these in the Robinson Crusoe Islanders, and we found a high proportion of South American variation, as you would expect, but not so much North American. So usually when you look in Chilean individuals, you see some of these North American influences coming down, but primarily we saw South American indigenous influences in the Robinson Crusoe population. We also saw, as you would expect, some Western European influences, so kind of Spanish, Italian influences. And again, this fits with some of the historical accounts that we have. And we also see some African influences within the genetic variation in this population. And we think that probably reflects some of the slave trade at that time. We also interestingly saw some Scandinavian genetic influences in the current population and some Middle Eastern genetic influences. So these don't necessarily fit with those historical accounts, but these nonetheless are quite strong genetic influences that we see in the current population. And I should say this is work done by my postdoc, Hayley Mountford. So she was able to map back from the current population to 23 founder individuals from 5 different families using the genetic data. And if we look wider than the mitochondrial and the Y chromosome haplotypes, we can see that the members of the Robinson Crusoe Island are about 50% European, 50% indigenous, and a couple of % African. So that's kind of how their genetic makeup looks to us today. And this largely resembles what we would expect of modern day Chilean populations. And this is quite good news for us because this indicates that the genetic contributions that we see in the islanders may well be relevant to other South American populations. So although they're a founder population and they're genetically kind of distinct, if you like, from the Chileans, there's still a lot of similarity between the islanders and current day Chileans. So I first became interested in this particular island, although the history is fascinating. I became particularly interested when this paper was published. Dorothy Bishop actually sent me a copy of this paper and said, this looks really interesting. You should look into this a little bit more. So this was a paper by Pia Villanueva at the University of Chile. And she described that the inhabitants of the Robinson Crusoe Island have a particularly high incidence of speech and language difficulties. So they estimated that 35% of the island children have what we now call developmental language disorder. So this is an impairment in the acquisition and use of language that is severe, it's persistent, and it's unexpected. So this doesn't come as part of a syndrome. This is an unexpected speech and language disorder. And in the UK, we would see between 5 and 10% of children have developmental language disorders, but on the island, this was much increased to about 35% of the children. They additionally reported that another 27% of the islanders had what we call secondary language deficits. So these are language disorders that were part of a wider syndrome or could be explained by other nonverbal deficits. So these are particularly high. And they also sampled the island children who were born in the mainland and had moved to the island since they were born. So these children didn't have any genetic links to the Robinson Crusoe Island. And among those children, about 4% of them had developmental language difficulties. So that's about the proportion that we would expect normally. There does just seem to be this increase in children who have this genetic link to the islanders. So I just want to take a couple of minutes out now to talk about language disorders, the kind of terminology that I'm using here, because I know that this may vary between the way that we refer to language disorders can vary. So here I'm using the catalyzed definition of language disorders. So here, a language disorder is a child who has language difficulties which create obstacles to their everyday life and that they are unlikely to catch up spontaneously. So these are not language difficulties that children then overcome. These are things that last with them and affect them day to day throughout their life. The term language disorder is a broad terminology that we apply in the UK. Within there will be a group of children who have what we call developmental language disorder. And as I said, this is something that can't be explained by any associated biomedical conditions. So something like brain injury or epilepsy, or some syndrome that perhaps might explain difficulties with language. if we see these comorbid disorders, then we tend to refer to that as language disorder associated with autism or hearing difficulties. And all of these disorders come under a very broad category that we now refer to as speech, language, and communication needs. Okay. So when I'm talking about speech, language, and communication needs, I'm talking about quite a mixed bag of distinct overlapping clinical categories. So this would include all of these acronyms here listed here, which you don't really need to worry about the differences, but all of these are slightly different syndromes or slightly different disorders that include some aspect of language difficulty, whether that is a primary medical concern or secondary to some other more pressing medical concern. And research in my lab, I'm particularly interested in are there similar genetic things that contribute to all of these, or are these distinct at the genetic level? So we do know that there's complex relationships within and between these categories. Children who have autism spectrum disorder will quite often have ADHD or other language difficulties and vice versa. So there's a lot of overlap at the behavioral level. But we don't really understand what causes each of these disorders, and quite often the best way to identify and classify these disorders. This is still a matter of debate. But the good news for me as a geneticist is that they are highly heritable. So when we think about genetic disorders, when I say something is highly heritable, then people quite often think of these kind of classic genetic syndromes, which are directly caused by changes to the DNA sequence. So you have a mutation in the DNA sequence, and that directly disrupts the function of a protein that is critical to the cell and critical to the development of the individual, and that is what causes the disorder. But these kind of genetic syndromes actually tend to be very severe and very, very rare in populations. There's also kind of this second class of genetic syndromes, which maybe don't quite fit with this classical etiology that people quite often think about. So these complex disorders, these instead of having a single mutation causing a single disease, here we're thinking about a balance of genetic and environmental factors which interact with each other to modulate your risk of developing a certain disorder. So if you think about something like heart disease, there are certainly classical genetic syndromes which directly cause heart disease, but there are, in the majority of cases, there are a complex interplay of lots of genetic factors, lots of environmental factors which modulate your risk over a lifetime of developing heart disease. And this is very similar to most neurodevelopmental and speech and language disorders. These tend to be more common in populations. So as I said, we would expect developmental language disorder affects between about 5 and 10% of the child population. So this is a relatively common disorder. And instead of there being a disruption of one critical protein, what we think is going on here at the genetic level is there is a dysregulation across these combination of genetic variations, a dysregulation of important pathways, which then increase your risk of developing a given disorder. Okay, so here there's really a distinction between these monogenic classic genetic syndromes and these complex disorders. And in terms of language disorder, we certainly know that there are monogenic or classical language disorders. So I think Simon Fisher has been to give a talk and he discovered the FOXP2 gene. We know that disruption of the FOXP2 gene will certainly cause you to have childhood apraxia of speech or some kind of neurodevelopmental syndrome. And there are, thanks to Simon's research and others, Angela Morgan, there are a growing list of genes that we know play a part in these language syndromes. So these genes, when you disrupt them, they directly cause quite a severe form of speech and language disorder, which is usually forms part of a syndrome and is quite distinctive in its appearance and relatively rare in populations. We also know from the research of others that there are such things as complex language disorders. So the majority of developmental language disorders, we think probably have this more complex genetic etiology. So there have been genes where there have been genetic variations identified in many, many genes, each of which explain a very small part of your risk of speech and language or developmental language disorders. And these tend to be in things like DLD or speech sound disorder, these more kind of complex and variable speech and language disorders rather than the syndromic severe forms. And I've just put links here to a couple of recent reviews if you're interested in reading a bit more about those two kind of ends of the spectrum there. So what kind of language disorder do the Robinson Crusoe Islanders have? We went and we collected information from 50 children who live on the island, and these came from 45 different families. We tested them across a range of language and nonverbal measures. And we found that 71% of the child population had a language disorder. So this confirmed what Pia Villanova had reported in her original report. Of these 71% of children, just over half of them had a language disorder that was associated with nonverbal IQ, and these tended to have a more severe form of language disorder. And the remainder of the children had that form of language disorder that we call DLD. So this tends to be less severe, and they have completely typical nonverbal development, but just these very specific speech and language deficits. We also found that there was a lot of what we call heterogeneity or variation between individuals. So it wasn't like individuals who have a disruption of FOXP2 have a very characteristic speech and language phenotype associated with them. But here we see great variability from one individual to the next individual in terms of the specific features of language that they struggle with. And this is typical of the kinds of features that we see associated with DLD in the general population, rather than these more severe forms of syndromic disorders. We also tested 107 of the first degree relatives of those children. So most of these were parents, but also some siblings. And we found that 58% of the family members also themselves had language difficulties. And those language difficulties were more prevalent in the members of the language disordered probands than those children who had typically developing language. So the relative risk here was 2.2. This says to us that if you are a family member of someone who already has language disorder on the Robinson Crusoe Island, then you are 2.2 times as likely to also yourself have language difficulties. Interestingly, we saw that this risk was consistent across the language disordered groups. So although those children who had language disorder associated with nonverbal IQ seemed to have a more severe disorder, the risk associated with language disorder was similar to their family members than to those family members of children with DLD. So this study indicated to us that the children of the Robinson Crusoe Island don't seem to have a very rare syndromic form of language disorder, but seem to have what we would refer to as developmental language disorder. The next step in the study was to start looking specifically at DNA variations. So in that previous study, we were simply asking who had a language disorder. Here we were able to collect DNA from islanders themselves. And Pia also did a lot of work to map back the relationships between islanders. And we found that 88% of the language disordered children could be related back to one single pair of founders who came to the island in 1867. So this essentially is one enormous family, which consists of 227 individuals over 7 generations. And the majority of language disorders individuals could be related back. The remaining individuals we think probably could be related back, but we're missing some of those critical links. So our hypothesis is that the individuals who first colonized the island had some genetic variation which put them at a high risk of speech and language disorders, and that has concurrently spread through the current day population. So there are 2 ways in which this could have happened. There might be that there is one single mutation that directly causes a speech and language disorder, and this has simply spread through the population. Or it might be that these original founders had a particularly high genetic risk across many, many different genes, and together in combination, because of the way that this population is isolated, this explains the high risk of speech and language disorders on the island today. So the first thing that we did was we took this enormous pedigree and we looked at the way that chromosomes are inherited from parents through to children, and said, are there any chromosome regions that we can spot that are shared between affected islanders? And we found the most commonly shared genetic region was on chromosome 7. And this immediately jumped out to us because this includes the FOXP2 gene and also the CatNap2 gene, which we know are important in speech and language development. So it's important to say that not all of the affected islanders inherited the same chromosome region here in terms of those original ancestors, but this can be quite complex to map out when the pedigrees get this big. So this was shared among more individuals than we would expect by chance alone. This is what we're asking at the genetic level here. So as I said, this is quite a big chromosome region. So this is the whole of chromosome 7, and it's about half of it that is shared between affected individuals. And this not only includes the FOXP2 and the CatNap2 gene, but also includes many, many other genes, 2 of which we were particularly interested in. This thiamine pyrokinase 1, and this encodes for an enzyme which breaks down thiamine. And this jumped out at me because there had been this study previously which showed that in infants who have a thiamine deficiency are at an increased risk of speech and language disorders. And this enzyme can be deactivated when there is a high fish diet. So I've always been interested and wanted to be able to go to the island and test the thiamine levels in the children, but we've never quite got around to doing this. But this is a nice example of how environment and genes may well interact to modulate individual risk. Of what's happening, but a more comprehensive screen. Did I drop out there? Okay. So studies such as that can allow us to look at the way that chromosomes are inherited, but more comprehensive screens can be done nowadays simply by sequencing the whole DNA, the whole genome. So from chromosome 1 right through to the X chromosome, we can sequence every single base. So we went back and we did this in the islanders, and we started off, rather than looking at every single gene, we thought we will just look at the FOXP2 gene and the CatNap2 gene and other genes that we know are important in speech and language. So some of those genes that I showed a couple of slides ago that current research has shown to be important. We screened these genes and we didn't find any rare loss of function variants that co-segregated with the language disorder. So there were no protein changes in there that explained the risk of language disorder on the island today. So the conclusion from this experiment was that wider screens were needed. So we went and we looked at the entire genome sequence or exome sequence, all 25,000 genes across 5 of the children who are affected by speech and language disorders. And in this was in collaboration with Simon Fisher and with Alex Heushen in Nijmegen at the Max Planck Institute. And we were really asking here, is there a single genetic variation that explains the language disorder that we see in the islanders? So we asked, can we detect coding variations that were present in all 5 of these children that have never ever been described before and are predicted to stop a critical protein from working properly? And to cut a long story short, no, we couldn't. So we detected over 47,000 variants per individual in that exome screen. About 17,000 of those were within known genes. About 8,000 of those changed the coding sequence of those genes. And actually 100 of those stopped the genes from working completely. but none of these 8,000 variations were present in all 5 of those affected individuals. We did, however, see 9 coding variations that were present in 3 out of the 5 individuals that we sequenced. So if you remember, I said the speech and language disorder is quite variable between individuals. So this allows for some flexibility in whether the affected individual is actually affected or not. And among those, these are the 9 variations here, and you will see that one of these is in the Nfxl1 gene. And this variant particularly interested us because we found that it occurred in total in about 11% of the Robinson Crusoe Islanders. If we only looked at those islanders who were related to the founder individuals that I've been talking about, then that frequency increased a bit. If we looked at islanders who were not related to those founders, then we never saw this variation. So we think this variation was probably brought across by one of those original founder members. If we looked in those children who had developmental language disorder, then we saw about 20% of those children carried this variant in the Nfxl1 gene. And if we looked in those children who had typical language development, we found about 5% of those children. So here we're seeing an enrichment of the variation in those children who have developmental language disorder. When we looked in European controls, we never ever saw this variation. But when we looked in South American controls, we did actually find it's present in South American individuals at a frequency of about 5%. And this matches up quite nicely with what we see in those islanders with typical language development. And again, there is an enrichment in children with developmental language disorder. So what does this variation do? This variation, as I said, is in the Nfxl1 gene, which encodes the nuclear transcription factor Xbox binding like 1. So this gene has this Xbox motifs that President Ono was talking about before. It's found on chromosome 4 and is a transcriptional repressor. So it turns genes on and off during development and primarily off during development. And genes like these are always really interesting when we think about neurodevelopmental disorders, because they're really important in setting up early cascades in gene expression. And we know there've been a lot of papers actually recently. We don't know much about this gene, but we do know it's important transcription factor in the differentiation of embryonic stem cells. So if we take embryonic stem cells, which are the very earliest cells from individuals, then in order for those cells to differentiate or turn into specialized cells within the body, then the Nfxl1 transcription factor is really important in this process. The gene's called Nfxl-like 1 because it's very much like the Nfx1 gene, which, as President Ono said, is important in the repression of these class 2 MHC genes. And this was interesting to us because some research by one of my PhD students at Oxford had found that there were associations with the DRB loci, which are these class 2 MHC genes in children with developmental language disorders. We know a bit more about Nfx1 than we do about Nfxl1. We know that it's localized in the central nervous system, in the nuclei, in Drosophila. And in Drosophila, if you knock this gene out, then they have subtle morphological abnormalities in motor neuron axon nerve connections. So these are kind of movement disorders. And we know that movement disorders can be important in things like speech and language through studies of childhood apraxia of speech and FOXP2. So we went on and we screened the Nfxl1 gene in children who had speech and language disorders from cohorts in the UK. So this is part of the SLI consortium. And we found 3 more potentially deleterious coding variants in this gene in 4 out of 117 children who we screened. So this is 3 times the expected frequency. This is, again, and not that these variations directly cause a disorder, but they increase the risk of disorder in these children. So we found a novel synonymous variant and 2 novel non-synonymous or protein changing variants. And these were all inherited from affected parents in these families. And they all occurred at highly conserved sites within this gene. Okay. So this said to us that this gene might not only be important in the Robinson Crusoe Island population, but it might be important in terms of developmental language disorder and in wider populations. I guess the million-dollar question is, well, what does it do and how does it increase that risk? And that's the work that we're doing at the moment. We know, as President Ono said, the Nfxl1 gene is highly expressed in cells throughout the body. So here we can see the expression level going from light blue to dark blue across various tissues in the body. And the Nfxl1 gene is at the bottom here, and the dark blue indicates that it's just highly expressed across a multitude of tissues. Nfxl1 also is expressed in many tissues, but at not such a high expression level. Interestingly, it shows quite a particular expression level in cerebellar tissue, which is shown by the red dots here. So before this line here, this is gene expression, the expression of the Nfxl1 gene prenatally in different regions of the brain, and this is postnatally on this side. So prenatally, it's highly expressed across a range of tissues, and then postnatally, it seems to be primarily expressed in the cerebellar tissues, which again fits with that kind of motor disorder that I was talking about with Nfxl1. And that persists throughout life. If we look in this resource called the Allen Brain Atlas, so this is quite a busy slide and I apologize for that, but what we're looking at here is lots of different regions of the brain in adult brain tissue. And this is cortical brain regions on this side of the chart, and core brain regions, brain stem regions at this end of the chart. On this color map, the green represents low expression levels and the red are high expression levels. And this is the Nfxl1 gene at the top here, and then the Nfx-like 1 gene in these bars at the bottom here. And what you can see is that there is a particularly high level of expression, and they show similar patterns of expression, particularly in the brainstem regions. And this red region here, or this region here, is the cerebellar tissue. So they show very high, both of these genes show very high expression levels in the cerebellum, in the cerebellar cortex here, but also in the hippocampus here. And if we look at other genes that show very similar patterns of expression, we can see some of our favorite neurodevelopmental transcription factors coming up here, like P10, see if I can see any more, BRAF, SOX, it was on this list somewhere here. So these are genes that have been identified for speech and language and for neurodevelopmental disorders through the investigation of things like autistic disorders and these more severe kind of syndromic speech and language disorders. My PhD student, Ron Nudell, looked at the expression of this gene, particularly in brain cells that were cultured in the lab. And he again confirmed that there is a very high level of cerebellar expression. But interestingly, given that this gene is supposed to be a transcription factor, so it switches other genes on and off, we don't see that it's located to the nucleus at all. So these are cells here. The nucleus is the hole in the middle, and the red is where we see Nfxl1 expression when we grow these cells in a dish in the lab. So what's really interesting is if this is switching genes on and off, it should be in the nucleus, which is where the DNA is, in order to gain access to those genes, but that's not what we see. And we think probably this protein needs some kind of activation trigger to relocate it into the nucleus, where it then turns genes off. And the $1,000 question at the moment is, well, what is this trigger? It could be something environmental, or it could be something to do with the time. So it might be only during early development that this protein actually sits in the nucleus. And we've actually just hired a PhD student who will start in October and be working with Esther Becker in Oxford and with Barbara Jennings and myself at Oxford Brookes. And she will be looking at the expression of this gene in cerebellar tissue in mice, right through from prenatal phases through to adult life in mice, and trying to identify what this trigger may be. So I just want to, how am I doing on time? Have I got 10 more minutes? Or?
Yeah, you're fine. No worries. Please go ahead.
Okay, so I just want to spend the last kind of 10 minutes talking beyond the genetics. So this is where we're at with the genetics. We know that Nfxl1 explains some, or we think that it's likely to explain some of the risk of speech and language disorders in the Robinson Crusoe population. It explains about 7% of the risk, we think, but there are probably lots of other genetic variations. There may be some environmental triggers, so things like the thiamine link that explains some of the other risk. So the question here is, well, how do all those risks interact, and what are those other genetic risks that we haven't yet identified? We've done whole genome sequencing in 10 of the islanders, and we're trying to link that together to answer some of these questions. And the work that the PhD student Gabrielle will be doing in October will hopefully answer some of those questions. This is some work that we did last year in collaboration with Maggie Snowlin and Charles Hume at the University of Oxford, and this was done by Carol Messer. We had an ESRC grant to visit the island and evaluate a language and reading intervention study at the school on the island. So this was a 30-week randomized control trial, and we recruited 68 island children who all attended school on the island. And we gave them 30-minute sessions every day in reading, language, and reciprocal reading. And the question was, could this intervention help overcome some of the genetic predisposition in these children? So at baseline, when we tested these children before they even started the intervention, we found that 28% of the children enrolled in the intervention study had special educational needs, 26% reported reading difficulties, and 3% reported language difficulties, which was surprising to us, given that this is apparently a main feature of the island population. And there were an equal number of males and females, and about 2/3 of the children recruited to the study had mainland parents, whereas only about 1/3 had islander parents. So this may explain why we didn't see such an increase of language disorders as we would expect. When we went and tested the children, we were surprised to find that the children had inconsistent letter knowledge. These are children from the age of 5, but right through to the age of 15. And given that Spanish is a transparent language, we were very surprised to find this. And we actually had to redesign some of our intervention sessions to include some letter knowledge training. The children in particular found phoneme blending and segmentation difficult, so rhyming games they really struggled with. They had a particularly slow word reading skill with low accuracy, and they had a good expressive vocabulary, but very little structure to their narrative and spoken language. So this was what we found at baseline. After the 30 weeks of intervention, we tested the outcomes of the intervention by forming 3 factor scores, one around their pre-literacy skills. So this was phoneme awareness and letter knowledge, one around their reading skills, and one around their spoken and receptive vocabulary skills. And we found that the intervention had significant effects. So these are Cohen D values, standard mean differences, particularly upon pre-literacy skills, but also significant effects upon vocabulary and reading, and upon reading comprehension. And we were able to see within this structural model that the effects of the intervention upon word reading and vocabulary mediated those longer term effects in reading comprehension in these children. We also went back and we retested these children at 9 months of follow-up, and we found that these basic skills were maintained, but the reading comprehension skills, so those more kind of complex skills, were not maintained across those 9 months after the intervention had been withdrawn. We also interestingly found that the baseline performance was lower for children, those children who had island or colonizer from those colonizing families. And these children showed the biggest gains across the intervention. So this was really promising news to me, because I think it shows that genetic propensity can become overcome with the right support, and that this kind of structured support can particularly help those children who are at a genetic risk if it's put in the right way. But we also found that it was those children who showed weaker retention when we went back at the 9-month stage. So I think all children, but particularly those weaker children, are going to require ongoing support to retain and generalize the skills that we're teaching during that intervention. And at the moment, we're extending this program in Sao Paulo in Brazil with the British Academy. Okay, so just to summarize, genomics can help us to understand some of the complexities of language, and this can be applied in many, many ways. So I've gone over lots of research here. In terms of the population structure, we've found that the Robinson Crusoe population are admixed, and we're able to infer some of that admixture within the current population. We were able to show that the language deficits on this island were highly heritable and familial, but there is heterogeneity between individuals. The genetic risks underlying the developmental language disorders that we see look like they're complex, and this seems to reflect what we see in other mainland populations. So the risk effects there may be genetic and or environmental, and there's likely to be interactions between those. And this is an example in the thiamine deficiency that I was talking about, but also in Nfxl1, which itself explains 7% of the risk. It would be interesting to see the genes that Nfxl1 regulates, do they also then play a part in that wider risk model? So we have yet to identify the modifiers or the mechanism of that risk. And just to end on a positive note there, the genetic effects themselves are not deterministic, but we can target interventions to help mediate some of the effects, the genetic effects in those language disorders. And just to say thank you here, I should say sorry before I say thank you, because there are so many people who I need to thank, and I usually miss 1 or 2 people off. So if your name is not on this slide, but you did play a role, I would like to say thank you anyway. This is my lab, Oxford Brookes here, and this is the Clara team. So a big thanks to everybody who helped us get that intervention underway on the Robinson Crusoe Island. We do a lot of work with Jean-Baptiste Cazier, with Simon Fisher, and with Louis Carvallier-Carmona at UC Davis, and a lot of work with the SLI consortium. And thank you to you for all listening.
Okay, well, wonderful. Thank you so much, Dianne, for a really inspiring and lucid talk, and a really fascinating ecological situation that you explored here to learn something. So my name is Michael Kober. I'm one of the leads for the social exposome cluster, and we're co-hosting this event together with the language sciences cluster. And just going to describe very briefly, I'm in my living room in Vancouver, wearing a gray shirt and brown hair with some gray showing up and black glasses. So it's hopefully accessible this way. I'll have the pleasure of hosting the Q&A session, and I see we're already having some questions and comments actually coming up in the chat. So I'll invite people as they appear in the chat. So Martha, you had a comment, I believe. Would you be kind enough to share it with the group?
I think Martha had to leave as she posted that comment.
Oh, I see. Okay, so maybe I read it just in case people. So it's wonderful for me to see the advances in this kind of research since the work that Werner Gopnik and I did in the early 1990s. I remember talking about it at the Max Planck Institute in Nijmegen, and people were very skeptical. And now Simon Fisher, of course, and his group are located there, and the work has grown and developed in complexity and sophistication. Thank you. Very interesting talk. So appreciate it, comment. I think it reflects very much what we all feel. Now, next, Elena has 2 questions actually. So would you be so kind to ask them personally, if you wish?
Hi, Dr. Newbury. My name is Elena Lopez. I'm both a geneticist and a developmental pediatrician at BC Children's Hospital. And I just wondered if you could provide some information in regards to the full-scale IQ in these kids and cognition.
Yeah, so as I said, when we tested, when we did the familiarity studies, we found about half of the children who had developmental language difficulties also had quite low nonverbal IQ. The information that we don't have there, I guess, is from a longitudinal point of view. It would be interesting to see whether the language difficulties precede the intellectual difficulties, or whether they're both apparent from an early age. And we don't really have that kind of viewpoint there. But certainly about half of the children do have quite severe nonverbal difficulties as well as developmental language disorders, whereas the other half of the children had developmental language disorders, but not the nonverbal difficulties. So the key feature, I think, is the language difficulties, whether or not they do co-occur with the intellectual difficulties.
But I'm wondering if you could create some endophenotypes there, those 2 phenotypes, ones with low nonverbal IQ. And because I was thinking in such a consanguineous population with such large areas of loss or heterozygosity that we are bound to pick up. The other questions was whether you looked at other genes may not be language related, but still, necessary for language acquisition.
Yeah, so in that very first study where we looked at the inheritance patterns, we looked at loss of heterozygosity and copy number variants in that study. And we didn't see anything that was found in all of the affected individuals that completely co-segregated. But as you say, it would be interesting to generate endophenotypes and to look at those in a more quantitative way rather than just simply classifying people as affected or unaffected.
Chromosome 7 is funny for language, isn't it? Because we also have the kids with Williams.
Yeah, I don't know what it is with chromosome 7, but
yeah,
we seem to keep coming back to it.
Thank you.
Thanks, great questions. Moving on to Dan, who not surprisingly is wondering about the cerebellum. Dan.
Yeah, thanks, Dianne, very much for that talk. I was wondering, with the finding of the Nfxl1 or Nfx1 and its predominant expression in the cerebellum, it was a bit surprising that one of the comorbidities that was not found, at least you didn't report it, with the language delay was a developmental coordination disorder. Could you speak to that at all?
Yeah, so we haven't found any evidence for developmental coordination disorder as part of that wider syndrome. That doesn't mean that some children on the island may not have that. I don't know. I was hoping that Pia might be able to dial in because she would be able to give more information about exactly what tests we've done. But it's not an apparent feature of the disorder. That doesn't mean that they don't have subtle cerebellar deficits. And of course, there's a kind of growing awareness about the role of the cerebellum in language in more general terms, as well as movement. So this is something that we're hoping to be able to tease apart in collaboration with Esther Becker with a forthcoming PhD.
Yeah. I know that it's a remote place, this island, so imaging might not be a possibility, but have you done any neuroimaging with this population to look at structural bases for the disorder?
No, we've talked about it several times, but that's another thing that we've never quite managed to do.
Yeah, thanks. And maybe as a lead into the next couple of questions, what kind of interventions have you tried and what has been the most effective?
So the reading and language intervention study that we did the year before last, that's the only intervention that has ever been tried on the island. And I think it was quite successful. The effect sizes that we were seeing were quite consistent and were maintained even after we left the island, but they are very, very resource heavy. We literally had to fly 4 researchers to the island, and they lived on the island for a year to carry out that study. So the work that we're doing in Brazil at the moment is trying to think, well, how can we scale up these intervention studies to make something more or less resource heavy and something more sustainable that can be applied to populations like this.
Yeah, cool. Thanks a lot.
Okay.
Right. Thanks, Dan. I'm gonna maybe, because of the intervention, maybe move to Jackie. MV has some questions regarding intervention as well before we come back to Janet. So maybe Dianne, Jackie was wondering whether you can share the details of your treatment protocol.
Yeah, so I can send you the paper, and that had full details, but so each child had one-to-one reading. So the letter knowledge work that we did was one-to-one, and the children were taken out for 30 minutes a day, every day where possible at school. Once those letter knowledge skills were in place, then we started one-to-one reading sessions with the children, and also very small group reading sessions where they would work through stories, and we would look at features of narrative, we would look at vocabulary within those stories and the way to kind of tell stories and the language that was used.
Thanks, Dianne. So I'm turning it over to a small group has a question. Please go ahead.
You're muted.
I don't know where small group is. Maybe I read the question.
She's there. She's there.
I got it.
Just muted.
Just forgot my mic.
Okay.
My name's Rebecca Dixon, and I'm interested as to whether the interventions and the persistence of the success of the interventions was affected by the age the children were when they started. I'm particularly interested because I'm a teacher of adult literacy, and I would say most of my students had undiagnosed language difficulties, and they're trying to make up for them now.
Yeah, so we didn't actually see an effect of age. The main difference that we saw was that those children who started at a lower level made the biggest gains over the length of the intervention, but we didn't see any age difference. But of course, we're looking at there were 34 in the intervention group, 34 in the control group, and they were spread across a wide age range. So it was hard to look. We didn't see any obvious effects, and we certainly didn't see any obvious effects of age in terms of that baseline either. There were some 13, 14-year-olds who had very poor letter knowledge, and there were some of the younger children who were starting to read. So it was quite a mixed group.
And I guess it would be probably too small to test whether any of the genetic variants affect the success of the intervention.
Yes, yeah.
Great.
It definitely wasn't as pure as some of the intervention studies, or as pure as Charles Hume would have liked in terms of the intervention, the design of the group. But at the end of the day, when there's a population of 600, you don't have so much choice.
Right.
And that's the real world application, I guess.
Vicky has a question. Please go ahead. Okay, maybe I read it then. So Vicky comments, it's a great presentation. I think we all agree, thanks. And she would like to know more about the Colombian control samples regarding the 042 frequency for the Nfxl. What parameters in this population was used?
So this was the Colombian population from the 1,000 Genomes project. I think, let me just check on the slides. So we have limited South American control populations that we can look at. There may have been 2 Colombian groups in there. Okay, can I just share my screen again?
Sure, go ahead.
Yeah. Okay, so with these, the CLM here are the Colombian individuals from the 1,000 Genomes Project. And these are Puerto Rican individuals and Mexican individuals. So they were the kind of control groups that we had available to us in terms of genome sequencing. But we also then collected our own Chilean controls from Santiago and our own Colombian controls in collaboration with Luis Cavalier Camana. And these were from a Colombian study that was looking at population controls, and they were looking at height, I believe, in population controls. But these were, so they weren't matched in any way. They were literally just a Colombian sample set that we could get hold of. And we consistently saw that the variants had a frequency of about 5% across all of those South American control populations that we could find.
Thanks, Dianne. I think that answers the question. Well, certainly, always challenging to find control groups for these unique populations there. To Elena has another question / comment.
Just, I'm from Mexico. So I just find some of these disorders, quote unquote, need to be studied more from a sociological point of view and how they shape culture and whether it influences this heritage of oral storytelling rather than the written storytelling. I find all of that really fascinating. From an evolutionary point of view, how we make maybe those genes a lot more present because, you know, the oral storytelling and heritage of oral storytelling is so important.
Yeah. Yeah, I think it's absolutely that I don't think you can tease those things apart, that it's just so, language is just so rolled up in our sociology and culture, as well as being a very useful tool in terms of communication. So yeah, I think that's fascinating.
Right. And the next question is from Barbara.
Hear me okay?
Yep.
Yeah, okay. And very interesting population that you're looking at there. My background, I'm a speech language pathologist and researcher, particularly in the area of speech sound disorders. I'm interested in, when you say that they had reduced structure of language when you did the testing of the cohort, I'm just wondering if you could give us a little more information. Does that include sort of short sentences with lack of complexity, no modifying clauses, reduced sort of variability in their vocabulary, deletion of morphemes, speech sound disorders? So what do they actually sound like?
Yeah, we didn't see so much morpheme deletion or speech sound disorder, but we certainly saw a smaller mean length of utterance, less complexities. So as you say, we measured the different clauses that children use. So we saw less use of those clauses and disordered in terms of the way that once they started pulling sentences together, they were disordered in the way that the sentences were organized. And the language that they used was quite restricted and not always used in the right way.
So, and these were the children. So in communicating with the adults there, do they speak similarly? Do they get to the point where they actually use more complex utterances, or are they still sounding like they're still speaking sort of laconically with not much elaboration?
So the older children in the intervention study certainly had similar language use to the younger children, and over the course of the intervention, all of the children were able to overcome some of these difficulties. But that's the only kind of longitudinal data that we have on any of the children on the island. Nobody's really studied the adults. We did a very quick language battery when we did the familiarity testing, but we haven't done any structural language analysis on them. But again, I think this is a really fascinating question about how does it shape the language disorders that the children have growing up in an environment where language disorder is so prevalent.
Yeah, so I guess my question, I mean, it's a sort of hypothetical future thing. Like if you were to work on the adults, then, you know, they would be providing a richer language environment for the children. You know, we always start with the children, but, you know, there's never any resources to deal with the, and talking about resources, I totally understand what you're saying. This is like way off the coast of Chile, and I can't imagine there's very many reading specialists and SLPs on the island at all. So that's complex. Thank you.
Thanks so much, Barbara. Now, Janet, you had some comments and some questions. I see that actually similar questions to the ones that I had. So why don't you go ahead?
Well, I'll just ask a couple. Barb just asked one that I wanted to drill into, so I won't go there. I was just really interested in the thiamine deficiency group talked about, because probably people on Robinson Crusoe Island maybe have a fish-rich diet. Does that provide some protection? And as they and perhaps other, you know, indigenous peoples are required to leave, or this is a more mixed population, of course, their traditional homes and go other places, and or just, you know, fishing gets worse, is that going to like increase the amount of expression of these disorders?
So again, this was just something that I thought was really interesting, but I have no kind of evidence to back this. So a high fish diet reduces the amount of thiamine. We're pretty sure that the children don't have a thiamine deficiency as it's described in the picture, because that's quite a specific phenotype that you would see associated with that. But it is possible that they have a genetic variation within that TPK1 gene, which, if that goes in hand with a fish diet, that together those two may reduce the level of thiamine, which during neurodevelopment may have specific effects upon their language development. And it's something that I would love to go and test, but again, logistically, it's just really, really hard to do.
I'm glad I asked the question because I had it backwards. So that's great. That's great. And I'll just do one more. Maybe this is what Mike was going to ask as well. You're going to be looking at expression in the cerebellum across the lifespan. You mentioned that it was a little surprising that you didn't see much expression in the temporal lobes. Is there any, I mean, I know one can't do everything, but is there any push to look at that across the lifespan as well, or perhaps, you know, thalamocortical or whatever? Yeah.
Yeah, so I think the things that we can do with the gene expression data sets are getting better all the time. So there's much bigger control data sets out there. We can do some kind of network analysis to look at what genes are correlated with Nfxl1. And once we have an idea about the kinds of genes that Nfxl1 represses, then we can look at those in terms of where are they expressed in the brain during development. And that's really where Esther Becker comes in, because she looks at the organism level. So she looks at brain development across the whole brain, from prenatal in mice right through to adulthood. So although she specializes in the cerebellum, we will be looking at those other tissues. And she also works with embryonic stem cells, which we can then induce into different neuronal types and look at the cell level as well as the organism level. So that's some really exciting directions that we can take that in.
I have a million more questions that relate to my own research and the intervention, but I'll email you those.
And if I may, perhaps then, you know, of course, I love anytime you mention environments, a sort of little signal went off in my own brain. And I was curious whether you had any thoughts on, for example, the role of things like the epigenome or the microbiome. I know Brett Finley was on the talk, whether there is any sort of indication that it might play a role in modulating in part the effects of the environment that you described so eloquently.
yeah, we don't have any direct evidence, but I'd be very surprised if it didn't. At a genomic level, we still look at each gene independently and each variant independently. And I really think that this is a key to understanding neurodevelopmental or neurodevelopment and genetic networks, is starting to think about gene-gene interactions, gene-environment interactions. And of course, that's not an easy thing to do, but I think that really is where the key is. Gene environment, thinking about epigenetics, and metabolomics and a million other things, but that is the real life situation, that we can't always just be thinking about here's a variant and here's the behavior. There's a big gap in between those two things, and that we have to simplify to get from one end to the other. But I think that's where it really starts getting interesting, when we start to backfill into between the gene and the behavior.
Oh, absolutely. Lots of room for further important discoveries.
Yeah.
Well, lots of positive comments and certainly really fascinating talk. Before I say goodbye, I just want to also thank the wonderful teams at both the Language Sciences cluster and the Social Exposome cluster, Ella, Alex, Ruan, and Kim, for doing such an outstanding job organizing this seminar. I want to again acknowledge and very grateful to President Ono for making the time to join us and sharing some of his history on the sort of gene. And foremost, I want to really thank Dianne for giving us such an inspiring talk. I think it was fascinating example of how we can use ecological situations to make important discoveries that are truly interdisciplinary and important for humankind. And really, the amount and quality of questions that talk elicited was a clear indication of how much thought and insight was stimulated. So thank you so much, Dianne. We hope next time we'll see you in person here in Vancouver.
Thank you for inviting me, and again, thank you to all the organizers, all those people who made it happen.
Thanks, everyone. Wishing everyone a good rest of the day and good evening to Oxford.
Thank you.
Thanks, everyone. Bye now.
Thank you.